Hi, good morning, everyone. It is a great pleasure for me to introduce our Plenary speaker. We have two here. So the first one is Prof. Elizabeth Aiken from University of Queensland. So Prof. Elizabeth obtained her bachelor in agricultural science, in crop science, from Edinburgh University, and a PhD is actually in forest pathology from Aberdeen University. And then after that, she became a postdoctoral fellow in the UK, and she worked on transposon tagging of rust resistance at Birmingham University, and also Sainsbury Lab in Norfolk. In 1993, she moved to Australia, to the University of Queensland, where she has been since. Okay, so much of this research at UQ is actually focusing on diseases of tropical crops, in particular banana, ginger, and sunflower. Okay, so the research topic has included the genetics of plant pathogen infection, molecular aspect of pathogenicity and disease diagnostics. Current research includes identification of resistance to Fusarium wilt in diploid banana lines for potential deployment against Fusarium tropical spore, TR4. Other than that, other studies also include analysis of mutative pathogenicity genes, including 6 genes in Fusarium oxysporum affecting bananas, strawberry, and ginger, as well as identification of cryptic plant pathogen species in ginger and sunflower. Okay, Liz has mentored numerous postgraduate students and has a well-established teaching portfolio in plant pathology at all levels, from first year undergraduate to coursework masters. He has an active commitment within the university in the area of research, integrity, and biosecurity. Okay, without further ado, I give the floor to you, Liz.
Thank you very much. Thank you very much, Fatima. You can hear me okay? I'll now try and share my screen. Hopefully it's going to work okay, but do tell me if there's any problems. Okay. Are you seeing the screen? I'll put it into presentation mode.
Yes.
Yeah, right. Okay.
Yeah.
Right. Thank you very much. Thank you very much for inviting me to talk today, and to the University of Malaya for hosting this, to Professors Yasmin and Jenny for inviting me, and to Fatima for introducing me. And Fatima, we had the pleasure of her spending her sabbatical in our lab here in 2018, I think it was. I had to look it up. But so much of what she's very familiar with this. So hopefully you're seeing the slides speeding ahead here. What we see here is the University of Queensland on the St. Lucia campus, in a pocket of the Brisbane River. And it's quite close to the central business district. But you might see here on the far right, a place called Eagle Farm. And Eagle Farm was actually the location where Fusarium wilt on banana was first recorded. That's not to say it was the first, that's where it originated, but it happened to be there was an amateur scientist. He was a medical doctor who first, Dr. Bancroft, who first noted symptoms of Fusarium wilt on banana. So, so this was back in 1876. So move on a few or over a century later, we're still working on this disease, and it's set up many problems for us. Here we see it's caused by Fusarium oxysporum cubense. Here in Australia, as in many places elsewhere, we see it manifests its problems under the name of different races. Now, race 1 was the one that caused all the problems on Gros Michel in the 1950s. It was also a problem here, and still continues to be a problem in Australia in certain regions. Some of you possibly less familiar with what is subtropical race 4. And we see that here in southern Queensland. It's also present in South Africa, causing problems on Cavendish in subtropical conditions. Suboptimal growth in particular is associated with the ability of that strain or race of the fungus to attack the plants. But tropical race 4 is the one that we're all becoming more and more familiar with, that causes problem on Cavendish and other cultivars in tropical regions, as well as potentially in subtropical regions. So just, and here we've got a classic, it's actually a ladyfinger plant, which is susceptible to race 1 and showing that typical collapse of the leaves and the yellowing of the leaf, etc. So if we look at the diversity of the pathogen that causes these different forms of wilt on banana, we find that race 1 and race 2 clusters together in a large clade, but interspersed within that, we have other Fusarium oxysporums attacking other plants, such as tomato, as cotton, etc. Whereas we see the subtropical and tropical race 4 cluster in another clade. They're quite distinct from each other, but they're again very distinct from the race 1 and 2s. And there's certainly moves at the moment to rename the Fusarium oxysporum in general and to divide it up into different species. It's an asexual fungus. It exists in not just these pathogenic forms, but in non-pathogenic forms as endophytes on a wide diversity of crop plants as host-specific forms. And we often talk about the banana infecting strains FOC being polyphyletic. If we look at the diversity of the pathogens using a system called 6 genes, these are the secreted and xylem peptides that were first identified by researchers at the University of Amsterdam working on the tomato infecting strains. And they found that these little peptides were secreted in host plants when the fungus was infecting them and moving up through the xylem, that's the name. We started to look at these patterns of 6 genes, they're all independent little peptides, and the sequences of genes that produce them. And we found that we could see a pattern amongst them of presence, absence, and diversity within the actual sequences of these genes, correlating with the different races of the pathogen. And in particular with tropical race 4, we could find some variants, diversity from subtropical race 4. And that became a useful diagnostic because distinguishing and knowing that you've got tropical race 4 present, etc. Sorry, the slides are advancing a bit quicker than I'm talking. So what we have here is the diversity of the fungus that we find in Australia. We find that traditionally I should say, race 1, it's been present from way back in 1876, as I said, in plantations in southern Queensland. It's spread up through the 20th century into different regions in North Queensland, where our main banana production area is. And then it's also present in Western Australia. Then we had subtropical race 4 start to become present in, it was observed in southern Queensland in the 1990s. And then also in the 1990s, we got tropical race 4 present in Northern Territory, and where it's expanded out through the Darwin region and affecting most of the banana production in that region. So we were aware of this. Now, having said that, 90% or more than 90% of production of banana in Australia is in the tropical north of Queensland, in the Tully region around there. And we were very worried about the emergence of tropical race 4 there, and it happened in 2015, if I recall rightly. It's still isolated to only a few plantations. They managed to, through good biosecurity protocols, managed to limit its spread. But the industry as a whole, which is very much based on Cavendish, is very concerned about its spread to other plantations. So just looking a little bit at Fusarium, what sort of symptoms it causes, how it infects. We've known about this pathogen for over 100 years, so there's a lot of background knowledge there. But we had a project where we were looking at a GFP transformed strain of subtropical race 4. I emphasize we don't tend to work with tropical race 4 here in Brisbane because of biosecurity reasons and the concern that the industry has of spreading the pathogen around. So much of our work is on subtropical race 4, which is already endemic here. So we looked at using a GFP transformed strain. We were able to not just look at the infection process, which a lot of people have done internationally, but we looked at its spread through the plant. Having said this, these were still pot plants in contained environments. And we found that the xylem was infested with the pathogen far in advance of symptoms being developed. And this had implications for trash and debris because you could go in and de-leaf in a plantation to try and minimize the consequence of pseudocercospora. In the meanwhile, that those leaves could still be carrying the Fusarium in them. And the important thing to note about Fusarium oxysporum, unlike most plant pathogens, it's an incredibly good saprophyte. So once the leaves are dead, the fungus would then colonize the leaves very quickly and produce chlamydospores or long-lasting spores, and the debris fall back into the plantation. Another thing we observed in just a quick study, just to go through that, is that when we actually applied fungicides and also herbicides, fungicides seem to have little effect on the control of Fusarium. And when we applied herbicide to an infested plant, it actually increased the inoculum again, because again, the fungus, taking advantage of that good saprophytic growth mode, was able to colonize the decaying tissue and produce spores. So again, this has implications for containment and control protocols in trying to eradicate an infestation of, presumably of tropical race 4 in a plantation. So what we left with, what can we do? It's so difficult to, hygiene is so important, but it's difficult to control once it gets in. We don't have effective fungicide against it. So, you know, breeding would seem the obvious option. And as Jim Lorenzen highlighted before, breeding is one of the main things that we can do to, not just for disease resistance, but for all those other agronomic traits that we want to enhance in banana production. But banana, as we know, is a bit of a freak. Sorry to say that, to a Congress dedicated to banana, but basically the banana that we cultivate only survives because we cultivate it. It's a sterile polyploid, parthenocarpic, and it's clonal. So, and obviously you know that the origins come from both the A and the B genome, acuminata and bulbiciana. But wild banana does exist. And this is an example of a rather skinny diploid. Here it's subspecies Malaccensis. And these diploid wild bananas produce seed. So, you know, it wouldn't be lovely if we could take up that option that Jim mentioned, that going back to the basics and breeding with these wild bananas to see what we get. Because the cultivated banana, for edibility, we have these undesirable breeding. If you want to look at them as breeding traits, they're totally undesirable: parthenocarpy and sterility. That's what makes it breeding that much more complex if you're trying to aim for that. But maybe better to play around with the diploids, with the seeded diploids first, and then go for parthenocarpy and sterility at the end. So in addition, in any breeding program, you've got the long generation time, you've got large size plants, and then you've got the complexity of interploidy crosses that leads to lots of unequal crossing over and issues with the progeny in such forced crosses. So let's just look at Malaccensis, Musa cuminata subspecies Malaccensis. It's a wild progenitor in many of the cultivated bananas that are utilized today. The fruits are small, they're seeded, and generally have no commercial value. and some of those individuals do show resistance to race 4 and also to race 1. And the advantage in our particular population that we also have individuals that are susceptible, and that makes analyzing the genetics a little bit easier. So looking at Malaccensis, we have the potential to help understand the mechanisms of resistance. So what we've done in this population, the initial population that we accessed through our colleagues in the Queensland Department of Agriculture and Fisheries, showed this segregation. It was an initial seed that was rescued through embryo culture and then planted out directly into the field. And we saw this wonderful, it was a field with subtropical race 4 was infested in the field. There was at that stage, there was no replication of the clones, but we did notice this lovely 3-to-1 segregation. And any plant breeder or any geneticist always gets excited by when you see something as clear cut as that. So we undertook self-pollinations, both of the resistant lines and the susceptible lines, and we've generated further populations doing that. We use embryo culture from the seeds, and as opposed to direct sowing the seed, and that allows multiplication of lines through tissue culture so that we get clones of each different accession. So that means we can test it in different situations, and we can also maintain the lines. For those of you who have never done breeding with banana, it's really quite fun. If you worked with Arabidopsis and you find those tiny little flowers, banana, one of the few advantages it does have, it has lovely big flowers and the monoecious. So you start off with female flowers in a spike as it develops, and then it changes. In the case of a Malaccensis, it switches across to the male flowers in about the 6th or 7th hand. And so you can get self-pollinations by taking pollen from one pseudostem to another, or you can do crosses. And so these are the little fruit we end up with the seeds, and we then usually embryo culture from those seeds. So with funding, we've been working on these lines for quite some time in the background with no significant funding available for that work. And then with funding from through the International Institute for Tropical Agriculture, who were in turn funded by the Bill and Melinda Gates Foundation on a project to look at improvement of banana for the smallholders of East Africa, we were able to bring onto the project Andy Chen. And Andy Chen has done a lot of the molecular work and also more recently the pathogenicity studies within our populations. So we've taken F1 of resistant plants, which showed a segregation for a single gene for resistance. So we've increased our population size so that we could start looking to see for molecular markers that associate with the resistant trait. And this is Andy Chen in the glasshouse here in Brisbane, along with one of the many students that we've had working on the project. So we initially did this bulk segregant analysis. We took the DNA from a collection of the resistant lines versus the susceptible lines within the population. And using illuminized sequencing, we looked at these two bulks, respective resistant and susceptible phenotypes. And it's quite notable what clear-cut phenotype we have. This is a pretty heavy inoculum, and sometimes the resistance show just a slight discoloration of the rhizome. It's of note that the fungus actually gets into the root very sporadically within the resistant line, but never progresses beyond the rhizome. In the susceptible lines, and this is 12 weeks after inoculation, we see a total breakdown of tissue, and then you can see the xylem discoloration up here at the base of that pseudostem. So taking, after quite a bit of effort, but just to sum it up, we located that resistance. It seemed to correlate with a region on chromosome 3. So it mapped to that. And we were able to do that because our Malaccensis line is closely related to the publicly available genome, which is based on reference genome based on, it's a double haploid of Pahan, which is also Malaccensis. It's not identical to it, there is quite a bit of diversity as we found out, but we're able to use the reference genome to help locate these markers. So, and we found it to be a region at the end of chromosome 3. So we then wanted to get fine map into there, because the idea was to get molecular markers that could be used not just for identifying the resistance genes, but to be used in breeding programs and interacting with the people at IITA and also with Embrapa and CIRAD as well. So this is sort of a complicated way to put it, but what we did is that we looked at a bunch of individuals within that population, which initially we would just screen for resistant, being the little red bars, only having next to no to zero rhizome discoloration, versus compared them with the individuals that were susceptible, having a range of rhizome discoloration up to quite intense, which we've illustrated here with the blue. Now, initially, when you look at that, we couldn't tell the heterozygotes from the homozygotes. But then we looked at a range of different markers, which we can see here as a band and a gel, because we converted them to PCR markers using identified, producing primers for them. And what we see here is if H is a heterozygote, we see in this case, we see all 3 markers. B is the homozygote susceptibles, where we see the 2 lower bands. And A, we could then work out retrospectively with the homozygote resistant lines. And this is a sort of procedure we've done. We've tried to get in a little bit closer each time by developing these markers. And obviously we're aiming for the ultimate marker, which will be the resistance gene itself. But at this point, we've used that marker. We've given it to our colleagues to utilize in Embrapa and IITA to see how it associates with the lines that they have. And we've fine marked some other markers. So the markers have been screened against genotypes in international breeding programs to determine if it associates with resistance. And the benefit of that you can do that in the absence of pathogen screening. You still have to do the ultimate pathogen screen, but we can reduce the numbers that you need to screen against the pathogen. And this is particularly important in a situation where you don't have tropical race 4, that you could screen it. Now, I should say that much of our work was initially done with subtropical race 4, but we take a subpopulation and we then screen them to see if we get the same response for tropical race 4. We can screen in the Northern Territory where they do have tropical race 4 present in Australia. So we doing phenotype assessments with tropical race 4 and also with subtropical race 4. We could look at a range of different cultivars and diploids that appear to be resistant to Fusarium, or those that are susceptible. The blue bars are just the controls. The orange bars show that the higher the bar means the more symptoms that were noted in these lines when challenged with tropical race 4. All these tests are done in pot trials, and in the Northern Territory, we did this in a screening house where the tropical race 4 is present. And we could see here that our Malaccensis, this is another marker band that we got, and we can see that upper band associates with resistance, as in a homozygote Malaccensis, whereas the lower band is on the homozygous susceptible Malaccensis. Now, we also found that marker in Madang and Pahang is a sort of a Malaccensis type as well. However, it was of note that there were still other resistant lines, such as Calcutta 4 and Pisang Jari Buya, which didn't possess that upper band, which didn't seem to correlate with our resistance band from Malaccensis. And when we've looked at other lines, we see that these bands seem to correlate with lines that have a Malaccensis as a progenitor, but don't necessarily correlate with other apparent sources of resistance. And 3362 is a line that was produced by the Honduran breeding program. And notably, we see it in our tests, it seems to be resistant, and we notice it seems to be heterozygous for this particular marker. And it's interesting to know that that line 3362 has both Malaccensis in its background as a progenitor, but also Pisang Jari Buya. So this took us to the next stage where we're interested to see is, could we find that there are other sources of resistance, or is it just that in these particular markers, they just, in that background, we're just picking up for the background of Malaccensis. So we don't know if the lines of Calcutta 4 and Pisang Jari Buya have an independent type of resistance, or if it's the same. So that's what we're doing at the moment. We're trying to determine if it's a different resistance. And I noticed that Jim Lorenzen mentioned in his talk that how important it would be to stack resistances. And that's what our aim is. It's good that we seem to be identifying this resistance in Malaccensis, but it would be interesting to see if the Calcutta 4 and Pisang Jari Buya resistance is separate, and if so, potentially we could stack these different sources into future lines so that they've got a more stable, durable resistance to Fusarium wilt in general. So we notice that Malaccensis is relatively close to Calcutta 4, but the interesting these is a wheel, I'd like to call it a phylogeny. This was produced by the Yaroslav's group at the Institute of Experimental Biology at the Czech Republic, and they do some beautiful work there. And they've looked at our Malaccensis line and seen where it fits phylogenetically. It's similar to Pahang, but not identical, as I said. It's Malaccensis in general is relatively close to Calcutta 4, but quite distinct from Pisang Jari Buya. So it's good that these may potentially offer alternative sources of resistance. So what we've been doing more recently, given that our susceptible Malaccensis is also related to Pahang, it's useful to use it as I call the universal donor, because our susceptible Malaccensis so far, we see it's susceptible to race 1 and race 4. And we cross it with these different resistance sources, ultimately get segregating populations so we can then start establishing where these other resistant traits are lying on the banana genome. So this is just some of the lines that we've been working with. In particular, we've crossed with Calcutta 4. I know that other people have looked at Calcutta 4 as well. It's incredibly useful line to work with. It's so fertile. It just produces an abundance of pollen. In our conditions in Southeast Queensland, the pollen is available all year round. We tend to use it as a male donor because you can get self-pollinations because of that abundance of pollen being produced. Sometimes the pollen production comes in some of the earlier hands, so we avoid that by using it as a male donor in a cross with a susceptible Malaccensis. When we've done that, we had a one-to-one segregation in the first F1 population from this cross, inferring that Calcutta 4 is heterozygous for resistance. And that seems to correspond to other studies looking at Calcutta 4 in regards to race 1. So that's quite interesting to look at that. And we're now going on to produce an F2 by selfing the heterozygotes to get a segregating population for using in mapping, where we'll get molecular markers there. Pisang Jari Buya is also incredibly interesting. It's a little bit not so easy to work with. It's a very, very tall plant. It's slow to flower, certainly in our conditions in Southeast Queensland. And we are limited by the accessions that have been imported into Australia so far. Bringing material into Australia can be quite take a long time because of the need to verify that they're virus-free. So hopefully we're intending to collaborate with Professor Yasmin on this and send our susceptible Malaccensis to Malaysia for maybe potential crosses with the Pisang Jari Buya present there. The line that we do have in Australia I was checking yesterday, does produce pollen, but very infrequently. So maybe we can still try do some crosses here as well using that line. But in the interim, we've crossed with the Honduran line 3362. which is quite fertile, surprisingly. And we've produced F1 from that. Initially, we see it segregates, although not in a huge population, so we're not 100% sure it's a single gene that we're dealing with at this point, but we're also generating populations there. It's unfortunately that population showing a little bit more of the Pisang Jari Buya traits, in that it's not as fertile as those from the Calcutta for descending populations, but we are producing some lines from it. So that sort of summarizes where we're going to, where we're at the moment. We're also looking at some other diploids and trying to do some crosses with a susceptible Malaccensis, potentially, so just scouring around to see if we can see other sources of resistance. But none of this work would have been possible without collaborations. And it was so interesting to see those your icons of Malaysian banana work research being discussed. All four of them seemed the big thing that they all did was collaborations, both at a domestic and an international level. And that is so important in banana. And we've been very fortunate through the Gates project that has opened up a lot more collaborations for us, but both internationally, but we also have good collaborations domestically. And we get funding from the Australian industry here, but also funding through the IITA, through the Bill and Melinda Gates Foundation. So most of what I've discussed today has been done by Andy Chen and other researchers in the lab. Liz Jaslowski's done a lot of the work with the 6 genes. Jay Anderson did the work with the herbicides, and Nolene Warman did the lovely work with the GFP. And we've had a whole array of students in the past working on that, including Fatima, who came and worked with us for a year on this project. So I'd like to, there's so many people to acknowledge, but I better stop there. Thank you very much.
Thank you very much, Liz. A very impressive shot of work being done in with you, particularly in Fusarium R4. So, well, is there any questions? So how's the progress now, Liz, on the breeding? Is it really impacted by the COVID?
Sorry, Fatima, I didn't hear that.
How is the work in Australia? Is it really impacted by the pandemic?
All right. Well, we're actually very fortunate in Queensland that some people might find it strange, but we closed our borders to New South Wales. And so actually we've had a few intermittent lockdowns, just lasting a few days at the most. Mm-hmm. I mean, last year was worse, but generally we've been able to proceed with our work as normal. And occasionally the university gets shut down, but at the moment there are no active COVID cases in the community in Queensland, although just across the border, 100 kilometers away in New South Wales, they are having quite a few cases. So at this point, we're able to proceed with our work. We can travel to North Queensland. We can possibly travel to Northern Territory at the moment, but we're just crossing our fingers that the vaccination rate will go up and that we'll manage to keep it at bay. Thanks for asking.
Next plenary speaker. So I would like to thank Liz.
Right, thank you very much.
Yep.